Good afternoon, and I hope you're doing good. So today, me, Hasti Abhiraj, will be presenting our research project on bird biodiversity and the urbanization effects. So our research project title is The Effects of Habitats and Human Involvement on the Diversity of Avian Species in British Columbia. Starting with the introduction, in which we have explained with the rising urbanization across Canada, various industries are on a steep incline to maximum productivity and consumption rates. With such rapidly changing sectors, many studies show rapid declining biodiversity in the forest across British Columbia. This research project argues if it is the urbanization of the habitat or the species favoring niches of the habitat which determines the bird biodiversity of the particular region. So our hypothesis was if the urbanization of a particular site has contributed any role in the bird diversification observed at a location, then the extent of urbanization could be considered as a contributing aspect of the region's bird biodiversity. The alternate and null hypothesis which we observed was if the habitat that is varied in accordance to different factors like precipitation index, migration patterns, age of forest, and natural impacts, including abiotic components like a fire and floods, would eventually lead to a decline in the bird biodiversity. Now, moving further, my teammates will explain the methods of the project.
My name is Hasti, and I am presenting the methods and results of the scientist bird biodiversity study. The scientists aim to compare the bird biodiversity between two distinct habitats, along with two control sites, so they can observe and distinguish better. The first habitat that was selected was Capilano University, being the highest urbanized environment, versus the McKay Creek, supposedly semi-natural habitat. From the observation that the scientists conducted using the Merlin bird app, they were able to record the bird species richness and Shannon diversity index, evenness, and average population size. Interestingly, the average population size that was observed for Capilano University versus McKay Creek were same because of the proximity distance between them. And now my partner will explain how we came up with the Shannon index calculations.
Hi everybody, my name is Aviraj and I'm here to explain you the calculations that were done in order to get the STI values that we have been learning for Capilano University and the McKay Creek. Before we go ahead, I would like to all of you to get familiarized with these terms. PI, that is the percentage in the decimal form, and the logarithmic function of the function that will be using in the whole calculations. In the second image, you can see elaborative data table that is considered as a raw data table. This table shows that we have done 5 trials of 5 minutes regarding each, where the individual species are being calculated, and one take represents only one bird here. So if you see the golden crown kinglet bird, it has 3 individuals in a total population of 30 species. After this, we move towards the calculative part, which shows us how we get the calculation. So the PI value, which is the 3 over 30 part, is equal to 0.11, and the logarithmic function of it is going to be minus 2.302. Multiplying these values of PI and the logarithm or its logarithmic function gives us the negative of the individual STI value that a species have. Of the individual species value, the formula of is the SDI value, Shannon in diversity index, is denoted by H, given by Shannon Weiner diversity index. We do the summation of all the negative values, and after the negative values we convert into the positive function, which generates an outcome of 2.4358. This 2.44 is really close to the calculation given out by the Omni calculator. That is one source that the researchers in this whole project used in order to find the individual uh, SDA values of one sampling size.
So now let's delve into the results. The results was incorporated using the Shannon Diversity Index, which stands for SDI, and the scientists used the SDI to measure the avian diversity. The SDI stands for the abundance and the evenness of species present in a specific habitat. And as you can see, for the results, the Shannon index, the SDI for the McKay Creek was around 2.0575, while the Capilano University exhibited a slightly higher average of SDI 2.2067, which suggests a greater bird diversity at the university. So, but in terms of evenness, when we use the Shannon index, the McKay Creek showed a slightly higher average value of 0.964 compared to Capilano University average of 0.923 in terms of distribution of certain individual species. However, Capilano University demonstrated a greater species richness. Additionally, the scientists want to suggest that the ideal data collection should have been taking 3 days of 3 trials of 5-minute recording in a 30-minute interval at a specific time in the AM and PM. So now we're going to focus on the results.
In conclusion, this research project tells how the urbanization has been affected, uh, being affecting the overall biodiversity of the region. This overall significance of findings of urban and the biodiversity conservation is really important. And in order to signify our findings, I can tell you about how the age of the forest that has been found in the lower conservatory reserve forest at the Rice Lake has been happened to increase the biodiversity of Capilano University. So if we are finding that Capilano University has a higher biodiversity, it means that the age of forest and the other features are also responsible for increasing biodiversity there. Hence, supporting a null hypothesis, or the hypothesis that we formed that urbanization will lead to has been actually not been true. And our findings has shown that the age of forest, precipitation index, other factors like the age of uh, the sleep cycles of birds, and the factors that have been told in the introduction have been a really important factor in determining the biodiversity at Capilano University itself, as well as this also goes for the Mackay Creek, uh, which is actually a secondary growth forest. And in order to see a lower biodiversity there, I think that the second growth forests uh, did uh, have been responsible.
We wanted to include that for moving forward for future studies. future scientists can address limitations that encountered in this study, such as data collection, and have a specific, time intervals, and also taking into account the factors that can influence bird population.
Where we actually have mentioned the books that have been used and the journals that have been really important, and also thanks to Dr. Paul Macmillan, which actually was the instructor through all this course, and his slides showed us how the other factors are being responsible for these. Also, I would like to thank Cornell Labs for their amazing Merlin app, as well as Omni Calculator that actually allowed us to analyze our results, raw data that we had. Thank you, everyone.
Thank you, everyone.